This is the first of two lectures that we're going to have in the Biophysical Techniques lecture series on introducing the wonderful world of NMR spectroscopy. What I'm going to do is look at the basic principles of NMR, where the NMR signal comes from, what properties of a molecule affect an NMR spectrum, and then we can work backwards from measuring an NMR spectrum. We can infer behavior of molecules in solution. And I'll look at the general scope of what's achievable within the biomolecular context. Right? Our NMR is a big subject, and we had decided to split it into two lectures. And on Tuesday next week, Jane Wagstaff will present a series of lived examples and working examples of how NMR can be applied in practice in this environment. Okay, so what I'm going to do is I'm going to start from very first principles, and I'll make no apologies. My personal preference is not to start from lots of heavy physics and mathematical formulae and derivation of where quantum mechanical rules are coming from. I tend to work deliberately from simplified cartoon-like pictorial representations of what's going on in the NMR spectrometer. But underlying that is a very rigorous scientific physical chemistry basis. You're not expected to pick up this kind of stuff yourself. There is an NMR facility out there with permanent staff who are there to help you in any way they can in planning out experiments, in implementing and running the spectrometers, and then in the interpretation of data right through to publication. So there are people there to help you. All you have to do is come up with the questions that we can answer with the various forms of spectroscopy. So where does NMR come from? What's the whole basis of it? We go right back to square one, and the basic premise that where you have a movement of charged particles, you have associated with that a local magnetic field. And this is very familiar to us in the form of electric motors. These are homopolar motors, and here we have some neodymium magnets in here and in here. If you pass a current through that wire, it generates a local magnetic field around the wire, which interacts with these permanent magnets down here, and it produces torsions and homopolar motor functions in these cases. But this principle also applies not just in wires in circuits, but also at the subatomic level, where you have subatomic particles that are charged in the nucleus and the electrons. As they move, they generate a local magnetic field. So inside every molecule, there is a landscape of local magnetic fields that is characteristic of that one molecule. And NMR is a technique in which we use these local magnetic fields to give us information about the composition inside the molecule, the functional group chemistry, the shape of the molecule, the conformation, how that shape changes with time, the dynamics, and also any molecular interactions and binding events and complexation that we see. So NMR is just exploiting these basic parts of electromagnetic induction to get the composition, the conformation, and complexation of molecules in solution. We have a well-equipped NMR facility at LMB. It's been around for 25 years now. And even before the LMB main building was opened, we had our own facility that is socially distanced away from the rest of LMB, about a 5-minute walk or so. And we house our NMR spectrometers in there. I should say that on a clinical site, it's unnecessarily alarmist to use the word nuclear magnetic resonance. If we had a big sign up outside the building, or if you have magnetic resonance equipment like magnetic resonance imaging, and you tell people you're going to put them inside a nuclear magnetic resonance imaging scanner, it causes unnecessary alarm. So on a clinical site, we always ditch the nuclear word and it becomes known as the Magnetic Resonance Spectroscopy Building or Magnetic Resonance Spectroscopy Facility. That contrasts it from magnetic resonance imaging, which is exactly the same technique that you get inside the clinical sites that are used for non-invasive imaging inside live human patients. Okay. So the facility that we have is at the moment changing its shape rather. We now have 3 high field machines at 700 megahertz, 600 and 500. I should say, for anybody who really hasn't come across NMR before, don't worry about this. It's just the strength of the magnet, and we'll come on to what these mean in a few minutes. We also have 2 routine instruments that support the organic chemistry efforts that are inside this building, and they're down on the ground floor in here. The model that we're changing to is that instead of having our own ultra-high field NMR equipment, it's very expensive and it takes up a huge amount of footprint of floor space. So you need large buildings in expensive sites to be able to accommodate this stuff. And the current model we're working on is for our ultra-high field work, we use communal external facilities that are at other parts of the UK, particularly the 950 and 800 megahertz spectrometers at the Crick. Also, there are facilities in Birmingham and other sites around. So that's the basic facilities. These are open to any of the projects in any of the divisions in LMB. You're welcome to come and use them. And as I say, we have a permanent staff of people for you to consult about exactly how to do it, should you not already have any experience in this. If you do come in with considerable experience in NMR and you decide you just want to run the spectrometers yourselves, that's also something that we accommodate, and we have people that do that. So why have people invested so heavily in all of this equipment? I mentioned a 950 megahertz machine at the moment. The most expensive NMR machine you can buy right now is around about £17-ish million for a 1.2 gigahertz machine. Why would anybody invest in that kind of equipment? It's a lot of money. So first of all, we get the structure. The NMR is one of the key techniques. Historically, it has been one of the key techniques for working out protein structure in the solution state. So crystallography has obviously had an enormous impact. It takes a photograph of molecules that are crystallized in a single state. We all know that proteins don't sit in a single rigid conformation. That's not how they work. Very often the mobility of the protein is instrumental as to the mechanism of action, how they recognize and how they catalyze reactions. So knowledge of the different conformational states that are present in the ensemble is an important aspect for us. So it's always been a good selling point for NMR that we can take images of the molecule in solution, and we also get the time dependence of any motions that are happening in loops, which can, in many cases, be clinically significant. NMR has been severely hampered by the speed at which it works. So if you have a protein that will crystallize, always the advice would be to crystallize it first, or if it's a large complex, get a cryo-EM structure of it. NMR is never going to be your first port of call for structure because it's very slow. It can take literally months. The last protein I did took me something, as a de novo structure, took something in excess of 6 months to do. But it was a protein that wouldn't crystallize, and it was too small for cryo-EM. So it's very valuable information. But what NMR is really slow at is coming up with candidate structures, candidate folded structures that you then assess with the NMR data. Assessing a candidate structure is actually quite quick to do by NMR. We're talking now, we can cut the project down to a few weeks rather than several months. So the advent of AlphaFold has been had a huge impact on NMR because it enables us to generate candidate structures that we can then validate. So we still have a role for working out from the AlphaFold structure, can we validate which of the proposed conformations are present in solution? And that's one of the roles that we fulfill. We look at molecular interactions and interfaces. So it is one of the gold standard techniques when you are looking towards when you have proteins in a pull down. Is that an actual direct interaction between those two proteins, or is it just all of them are part of the same complex? So we can get information on the binding interfaces. Exactly where do you bind these two proteins? Where would you be looking to put a ligand, for example, to prevent the association of these two proteins if that's what you wanted to do? We can do modeling of complexes in high detail using data-driven, NMR data-driven docking algorithms. One of the themes I'm going to come back to again and again is the key advantage that NMR has over other forms of spectroscopy. We see a separate signal for every atomic nucleus in the molecule. So we have atom-specific resolution of what interactions are occurring at what time. So we can look at things like protein folding, and that's what enables us to characterize binding interfaces. But this idea that we have site-specific resolution throughout the molecule is extremely useful. It provides information that no other form of spectroscopy can get a handle on. We can look at radius of hydration and diffusion, how compact or otherwise, how molecules can change their compaction can be done from hydrogen exchange rates and measurement of hydrodynamic radius. NMR is quantitative, so we can use the data we acquire to produce physical constants, measure physical constants in a way that is, again, with very high accuracy and on specific sites. So we could look at a specific glutamate residue, for example, and look at what pH does, or what is the pKa of that specific residue in the context of all of the rest of the protein as well. We can pull out with high resolution. NMR is used ubiquitously throughout the pharmaceutical industry as one of the key techniques for identifying ligand binding. So in lead compound generation in the pharma pipeline, NMR has played a major role for nearly all of the major pharmaceutical companies at some point, and it's still one of the techniques of preference for working out ligand binding. We shouldn't get too bogged down with the idea of just looking at the spectra of the macromolecules. We can also get a lot of information by looking at substrate turnover, and we can observe the substrate itself rather than having to observe the enzyme. That would be rather complicated. So we can do experiments in which we're looking at ligand binding for lead discovery. We can do it as metabolite turnover inside whole living cells. We can do enzyme substrate turnover. But the idea that we can look at small molecules to get a lot of information about how enzymes are behaving is a crucial point to get across. Okay, I should say that NMR, I believe, is changing its focus rather at the moment, and there are fewer and fewer de novo structures being entered. This is statistics from the Cambridge crystallographic database. The first NMR structures came in about 1989 with the introduction of labeling with the stable isotopes N15 and C13, and it enables us to get high resolution structures of quite decent sized proteins. The number of structures deposited per year peaked just before 2010, and it's tailed off since then. So we are redefining the way in which NMR is used in a biomolecular context. We wouldn't sell this primarily as a structure technique by itself. If you compare it with NMR, just as a comparison, compare the NMR with the depositions for X-ray structures, which are starting to tail off. And of course, the new technique on the block is EM, which is at the moment rising exponentially. So what we have to do as NMR people is push across the alternative uses for what this technique will do. The sort of thing that I'm doing, for example, is looking at multi-enzyme or multi-protein assemblies using NMR to look at isolated pairs of domains. The problem we have is as we go up in molecular weights, the quality of spectra and the complexity degrades quite quickly. So NMR is only ever really going to look at small protein assemblies. So it's highly suited to looking at individual domains and pairs of domains, interactions of isolated domains, rather than the entire complex by itself. So this is the sort of thing that we're looking at. We're trying to characterize which of those partners are direct binding interactions. We can identify their contact surfaces, and in some cases, we've done ligand screening to identify small molecules that will interrupt the binding. We can look at complexes either if they're stable or transient. It doesn't matter to us. We can still work out where the interaction surfaces are. But we should also emphasize that it is as part of an integrated structural biology effort, and we link into the information that we can obtain from other structural techniques as well. Okay. So where does NMR really start to struggle at the edge of its scope? Well, one of the first areas of a problem for us is the symmetry of molecules. We find it very difficult to resolve different planes of symmetry. So if you have, for example, a cholera toxin, which has a homopentamer of 5 identical subunits, you can't distinguish between the 5 different subunits in that homopentamer. So NMR is, however, the information that it provides can be really useful. That symmetry information can be exceedingly useful in working out what the structure of something is. So for C60 fullerenes, for example, in the carbon NMR spectra, you get just one signal, which tells us that all 60 carbons in that molecule are in an identical environment. So in constructing the structure of this, you have that constraint that all 60 carbons have to be perfectly symmetrical. So the information that symmetry is there can be quite useful, but NMR isn't going to give you individual peaks for the individual 60 atoms in them. In this case that's more relevant to our kind of work, this is something from Marianne Bientz's work. This is a homodimer, and one element, this helix element from one of the proteins, swaps across into the other. It's a domain swap protein, and it's a homodimer that is symmetrical, and we find it very difficult to see the 2 halves of that independently. We have strategies in which we can try to break up the symmetry using isotopic labeling, but it's one of the areas where NMR struggles a bit. Heterogeneity. NMR needs very pure samples, so you really need individual species in the solution. We require them ideally to be pure, highly pure, and monodisperse. It actually doesn't matter so much if it's a single conformer or not, because one of the things that we can pick up by NMR is the existence of conformational exchange, and we can do something to delineate the contributing conformers in the ensemble. Sample quantity is always an issue. For reasons I shall come into in a few minutes, NMR is a very insensitive technique, and it requires relatively large amounts of sample. This is the minimum requirements that we would have, is about 1/4 ml or so of at least 20 micromolar protein sample. The volume is fixed, and therefore it places a restriction on the minimum concentration. High molecular weight, as I've said before, NMR struggles with slowly rotating rigid molecules. As we go up in molecular weight, the quality of the signal degrades quite rapidly. So we start to struggle as we get to proteins that are high molecular weight, but we restrict ourselves generally to looking at single domains rather than multi-domain assemblies, or even intact whole proteins. Where we have a low spin density, which is really specifically referring to nucleic acids, in proteins we have a high density of NMR active spins, NMR active nuclei. Inside nucleic acid, the density of those is really low, and so we have very little information from which to build structural information. So where we have low spin density in nucleic acids, it's a lot more difficult a problem, but it's not something that's intractable. We just have to go to different techniques to use it. And the speed we've alluded to already. The data analysis can be slow. If you can crystallize it, crystallize it. If you can get it by cryo-EM, do it by cryo-EM. If you can do neither and you really want to know this information, there are ways of getting it by NMR spectroscopy. Now, this illustrates one of the issues that we come across. In crystallography, we crystallize out single snapshots. So we get a photograph of a single snapshot of a protein in a specific state. If you're lucky, you can find conditions and mutations that will trap the molecule in one or more of those specific states. But what you get is a static snapshot. We know that what's happening in practice is we have a high degree of movement inside the protein. What NMR sees is an ensemble of everything that's in the solution. So we have to use it in combination with computational techniques to delineate what are the individual likely contributing conformers. And if you ignore the fact that this is a dynamic assembly, what you can interpret is an erroneous virtual conformation, which isn't actually physically populated in solution, but it's the apparent conformation that comes out of the data. You have to be really quite cautious in the way that you interpret dynamic movements, but we are able to quantify the extent of motion and the rates of exchange that are going on inside the solution. This is something that I've nicked from Jane's talk that comes up next Tuesday. And it's where we've decided to split the talk, is there are a lot of details that we can prize out of a molecule about its dynamics. And this is the sort of processes that are occurring inside a protein and the sort of timescale on which these motions occur. And here is a range of NMR approaches that we can use that are quite specialist, and it's beyond the scope of a first introduction to go into each of these, but Jane will illustrate some of those next Tuesday. But the idea is there are different processes going on inside that we can get a handle on what motions are going on inside the solution. So the basic starting point of NMR is that we take an NMR active nucleus. Now, certain nuclei have an active nuclear magnetic dipole, a simple north and south pole. And as we put that nucleus inside a strong external magnetic field, we will get a preferred alignment of molecules of these nuclei, either with or against the applied magnetic field. So we are with or against the applied magnetic field. So what it produces, because of the quantum mechanical selection rules, what we tend to see is we only get these two states. We either get aligned with or aligned against the magnetic field. And so we get two different energy states in there. We get a lower energy state that is with the dipole aligned with the magnetic field, and a higher energy state where it's aligned against. Now, the difference in energy between these two states is what we're going to exploit in the NMR spectrometer. It's linearly related to the magnetic field strength. So as we go higher and higher in field strength, the energy difference between these two states increases, and we get an excess population of spins in the lower energy state compared to the higher. That's referred to as polarization. And it's the polarization, the difference in energy level populations, that gives us our energy, our NMR signal. The number of spins that we have in the lower state in excess over the upper state in our spectrometers is about 40 atoms, 40 molecules, 40 nuclei in a million, right? So we have only a very small excess population in the lower state. And that's why NMR is fundamentally a very insensitive technique, because we don't use the potential of all of the spins in there. We only have a 40 parts in a million excess in the lower state. So as we have NMR active nuclei in the molecule, they separate out. The energy level between those is directly proportional to the applied field strength. So we have NMR active nuclei for hydrogen. We refer to that as proton as well. I'm so used to referring it to proton NMR. A hydrogen nucleus, H1, it has no neutrons in it. It's just a proton, right? So we refer to it interchangeably as hydrogen NMR or proton NMR, okay? Also at very high natural abundance is phosphorus, the isotope phosphorus 31, which is almost 100% natural abundance. If we want to bring in isotopes of nuclei of carbon and nitrogen, then they are present at very low natural abundance. The only way we can bring these into play in the NMR is that we put these as they're stable isotopes. We can label the protein by growing the protein in a growth medium that contains C13 glucose as the only carbon source, and N15 ammonium chloride as the only nitrogen source. And then we have a much greater scope in which we can expand the range of observations. The other one that we use quite commonly is substitution with deuterium, but that's used in the opposite sense. Now, not to make it visible, but that can make the proton NMR invisible if we exchange away some of these protons for deuterium instead. We can make that portion of the molecule NMR invisible, which there may be applications for that. There are no NMR active nuclei that are useful for oxygen or sulfur. Other inorganic ions that are problematic is sodium or zinc, for example, that we have difficulty seeing. You can use surrogates instead, NMR active surrogates. So you can use rubidium as a surrogate. There's an NMR active form of rubidium used as a surrogate for sodium. And cadmium is used as a surrogate for zinc because cadmium 113 is NMR active. When you put the molecules into solution, all of the nuclei of the same isotope will resonate at exactly the same frequency. There is nothing else to tell it to resonate at any other part. Because of the quantum mechanical selection rules, it's extremely invariable, and we get absolutely identical frequency in the absence of other magnetic fields. The relative merits of going to a higher field strength, well, as we go to higher field, we get increase in sensitivity. The signal dispersion, you can see increases as we go to higher field strength. So for example, this is looking at sucrose from the sugar bowl, and we can see how the signals spread out their frequency in absolute terms as we go to higher and higher field strengths. Everything becomes a little bit more resolved from each other. You can see spins in here are very overlapped, and we're starting to resolve them out as we go to higher field strength. So this is worth the additional money for us. And as we increase the field strength by about 100 megahertz, it doubles the cost of a spectrometer. So you really need to have justifiable reasons for wanting to go to higher field strength, because it's huge relative cost. We also get favorable relaxation rates, which we may come on to as well. The disadvantage is it costs a lot of money, and the footprint for housing these becomes a huge problem. You've got to find the real estate to put this stuff on. The power handling, as we go to higher spread of frequencies, we have to put in more power. And so we start effectively microwaving samples as we go to higher power levels. And there's some accentuated line broadening, which I won't elaborate on very much. So one of the things that we have adopted, though, as a universal convention is the chemical shift scale. So that data that I showed on the previous slide, we had that expressed as absolute frequency. In the chemical shift scale, we express everything as a ratio to normalize the frequency against the applied magnetic field strength. In other words, every spectrometer in the world, no matter what its field strength, will have the same chemical shift for the center frequency of each of these NMR signals that we're seeing in there. Okay. So this is the idea. You'll hear me talk about the term chemical shift to depict frequency, which is just the frequency normalized against the field strength so that everything on every machine is the same. This is the business end of an NMR machine. It's the detector coil, and it's configured so that we can detect magnetization that is in the horizontal plane. It's rotating in the horizontal plane. We don't normally see this part of the spectrometer because it's buried inside the middle of the magnet, which is providing the large external magnetic field. The standard sample conditions that we would have is normally we go for a 550 microliter sample, which in this 5mm tube gives us a 4 centimeter sample depth. And the concentration range we would normally recommend is not going below about 20 micromolar, but also not going above 500 micromolar. If you do go too concentrated, you tend to see increased amounts of aggregation of proteins. You increase the risk of your protein aggregating. So we normally recommend not going above 500 micromolar, just as a rule of thumb. The temperature range is normal sort of solution conditions for aqueous solutions. Now, the coil, somewhere, there we go. The coil is set up to detect magnetization that is precessing, rotating around from these individual nuclei. They all co-align and they produce a net magnetic moment that is measurable in the receiver coil. Okay, so that at equilibrium, that magnetic vector is pointing straight up the bore of the tube, and we can't detect anything right inside the coil. So we give it a short pulse of energy that rotates that magnetic vector into the horizontal plane, and whilst in that condition, it rotates and it generates a current, a small current in the receiver coil. That is the NMR signal that we see. It's an oscillating sinusoidal wave. So we then have to convert that to the frequency, and the process is not complicated, but a little too lengthy to explain today. But we can take that oscillating sine wave, we put it into the Fourier transform algorithm, and what it gives us is the frequency of the wave that's coming from the solution. It is a delta function. It has infinitely narrow. It's just defining a single frequency point. If in practice we get that, this is exactly what we see. We have something that we excite the spin into the horizontal plane, and it then will relax back towards its equilibrium position. So it will be an exponentially damped sine wave. If we do a Fourier transform of that, what we have is a so-called Lorentzian line, and the center frequency of that is exactly the same as you would get for the infinitely long cosine wave. But in this case, we have, in addition, a definition of a line width. The uncertainty, if you like, of the frequency of that, which is a function of the exponential time constant. So this value T2 in here is referred to as the exponential time constant for the damping of that sine wave. And the width of the line is inversely related to T2. In other words, in simpler terms, as this signal decays faster and faster, the line width will get broader and broader. So if we look at this NMR signal, the time domain signal for a macromolecule, a protein, for example, what you see is very rapid relaxation, and it returns back to equilibrium very efficiently. Small molecules, on the other hand, the signal lasts for a great deal longer. So in other words, they're just depicting two decays that vary in this value of T2. And as we pass them through the Fourier transform, you see that the line width is very much broader for the macromolecule, and this is why NMR suffers as we get to higher and higher molecular weights. As we go up and up, the molecules tumble slower and slower in solution, and the values of T2 get shorter and shorter, and the line widths become too broad for us to make any resolution of the signals in them. So I'm very impressed with Andy Jerome's book from the 1980s. This is the ABCs of this don't change even from them, but it's a lovely analogy of what's going on if you haven't come across this technique before. The idea is if we take a resonator, it's depicted here as a bell. Bells have overtones and different harmonics. And in this case, in this analogy, we have a bell that is a simple harmonic oscillator that has a single resonant frequency, okay? If we take that bell, we put it in a box and we shake it up, what we would see is a decaying sinusoid, and we can then pass that through a Fourier transform and extract what the frequency component of that is. Really nice and simple. If we put another bell in the same box and we shake it all up, you get a summation of those two signals as your cacophony. You can put that through the Fourier transform, and now you can extract both frequency components that are in there. And you can keep adding more bells to the box, and you just get a more complicated cacophony. But as you pass that through the Fourier transform, at the end, you get all of the individual frequency components of the individual resonators. So that's a brilliant analogy for what's going on in Fourier transform NMR. We excite all spins in the molecule at the same time with a single burst of white light. It's all in the radio frequency range of the spectrum. So we get a single radio frequency pulse. We record the signal that comes off in the receiver coil, Fourier transformation, and hey presto, you've got an NMR spectrum. If we look at that for a 10 kilodalton protein, this is very typical, a nice small folded domain. So it's something a little bit larger than the size of ubiquitin, for example. So it's around about the size you'd get for individual domains in proteins. And this is just part of the NMR spectrum. It's showing the region of the amide hydrogens in the backbone of a protein. And you can see the immediate problem is there are so many signals in there, and the amount of overlap that occurs makes it difficult for us to extract anything really of genuine interest from a spectrum like that. So what we can do is we can spread the signals out into a second dimension. The physics of how that's done is beyond what we can go into today, just to take away the message that this is actually the same spectrum of the same protein. The only thing that we've done is each of the signals in here now is being separated in the second dimension by the chemical shift of the directly bonded nitrogen. So these are HN protons, right? And this axis is showing the nitrogen chemical shift for this amide. This axis is showing the hydrogen shift. That's quite an awkward representation for us to look at. It's much simpler if we look at it as a contour plot, looking straight down. Again, this is exactly the same spectrum. We're just viewing it in a different way. And in this one, we are looking down on individual spots. So for this representation, we're seeing an individual spot for every residue, every amino acid residue in the protein. And we can resolve almost everything. There are very few areas of overlap in there, and we get separate peaks. So we have an ability to examine the protein structure at each individual site. I will pass very briefly over this. The idea here is not to show how complicated NMR is, it's to show how easy it is. And it's showing the nuclear spin Hamiltonian for is spin angular momentum of an atomic nucleus when you put it into a magnetic field. And it's not the complexity of the expression, it's the fact that it breaks down into the sum of three simple terms that we're focusing on. Okay, so the first term here is referred to as shielding. The second term is J coupling, and the third is dipolar coupling. Okay, and we'll just briefly look at how these different terms contribute to the spectrum, and then how we can use those to interpret what's going on inside molecules in solution. So the basic premise is when we have a cloud of electrons immediately around any nucleus, it generates a local magnetic field. As these charged particles move, they generate a local magnetic field, and it will always oppose the applied magnetic field. So what we refer to is the external magnetic field is, sorry, the nucleus is shielded from the external magnetic field by the magnetic field of the electron cloud. So this gives us an enormous amount of information about electron density, so functional group chemistry, and it enables you to ascertain a lot about what the functional group chemistry is within a molecule. But it gives us a single frequency dependent upon what the amount of shielding is in around that particular nucleus. And we can say the chemical shift axis there we've already touched upon. The frequency is depicted as chemical shift, which is normalized, the frequency normalized against magnetic field strength. So in any small molecule or even larger, we have this landscape of local magnetic fields, and they are characteristic. They depend on the functional group chemistry, the shape of the molecule, and the time dependence of that shape, the dynamics. And these external fields contribute that each of the different nuclear sites in there are going to have a different, they're experiencing a different local magnetic field, so they will all spin at a slightly different frequency. Remember, in the absence of external magnetic fields and additional magnetic fields, all nuclei of the same isotope will resonate at exactly the same frequency, but in practice they don't, because inside the molecule there's this landscape of local magnetic fields that is a characteristic. We do also see around pi bonded systems, we see delocalized movement of electron clouds around carbonyl groups. You get this so-called shielding cone where you get enhancement or suppression of the magnetic field. So you get a whole range of effects in there that help the different spins to experience different local magnetic fields. J coupling is an effect that is transmitted through covalent bonds. And what we see is this spin experiences two different environments that are dependent upon the orientation of a neighboring spin, which is what we would refer to as coupling. Coupling simply means, in this case, the state of this system is dependent upon the state of a neighboring system. It's this local magnetic field coming from paired electrons, oriented electrons in the covalent bonds, and they give us some ability to connect between adjacent spins that are covalently bonded. So in fact, we don't see a single peak, as we just saw before, for each of these systems. For this one, for example, we would see two different signals that are separated by the amount of this local field that's in them. It's referred to as the J coupling, or scalar coupling. The third coupling effect is referred to as dipole-dipole coupling, and I think it's a little bit more intuitive. As you get two spins that have their magnetic moments, this spin will experience two different environments due to the direct interaction of the magnetic dipoles. This effect can be observable up to about 7 angstroms. So we're talking about 4 or 5 carbon-carbon single bond lengths. that sort of distance over which we can see the effect happening inside any molecule. Okay, that's the basic premise of the contribution of these. So in fact, the separation of these two lines is not just J, it is J plus the dipolar coupling field, but dipolar coupling has a property where it averages away to zero. The splitting averages away to zero as the molecule rapidly reorientates in solution. So as the molecule spins, the average value of D is zero. So actually the separation will be J plus D, but D equals zero. When we put a spin into solution, it has an orientational dependence of this bond vector. So this is looking at the dipole-dipole coupling term now. It has an angular dependence. The angle that that vector makes with the applied field strength is a factor that affects the value of the observed field, dipolar coupling field. This field, it averages away to zero. That doesn't mean it is zero. It just means as a time average, it goes to zero. And we can use the fact that it's present still, this field is still there, from measuring the value of these dipolar coupling constants. And it has this angular dependence here. It also has a distance dependence. Excuse me. If we don't average these spins and their orientation over time, so in other words, if we have a solid state sample where all of these molecules are just sitting in their own orientation, what we would see is that we resolve all of the dipolar couplings in all possible orientations. This is the typical sort of dispersion of frequencies that we would get in the solution state NMR spectrum, where the dipolar coupling terms have disappeared. But in the solid state, we lose all fine detail in that spectrum because it's dominated by these massive dipolar coupling terms. So solid state NMR becomes a very different technique. We don't do any of solid state NMR in-house in LMB. If we have any projects that need that, we collaborate with people outside. And the reason is it's a really very different technique, and it's dominated by the need to suppress these dipolar coupling terms. So the dipolar coupling gives us this ability to measure the bond distance in this particular case, and it comes in the terms of a parameter called an NOE. The NOE stands for nuclear Overhauser effect, and basically it's an interatomic ruler. It gives us accurate estimates of what the distance is between two spins. If we have a pair of spins for which that distance is fixed, so let's say the dipolar coupling between a hydrogen and its directly bonded nitrogen, we know what that R value is. So that comes out of the term as a variable. And what we get is the ability to measure the orientation of this bond with respect to a global diffusion axis. And this is referred to as a residual dipolar coupling. It just gives us the ability to measure the orientation of a bond with respect to the global diffusion axis. So this is where Kurt Wüthrich came in the early 1980s. He got a Nobel Prize for NOE-based structure determination of proteins. When you look at a protein and you superimpose on there the position of all of the hydrogens in there, you can see massive amounts of hydrogens. If you go to the process of assigning the frequency, the identity in terms of the frequency in the NMR spectrum of all of those hydrogens, you then measure pair by pair interactions between spins. You can get enough data to get a unique fold for the protein as it sits in solution. And that was pioneered in the 1980s. The structure is determined from short-range interactions. And we measure about 25 different spin-spin distances per amino acid residue. This is the raw data that we see in the NMR. It's that two-dimensional NMR depiction again. It looks quite daunting. It is quite daunting. If you have a peak there, for example, what it's telling us is a proton that has a spin frequency at that frequency is close in space to a proton that is at that frequency. If we know what the frequency assignments is for all of the protons in the spectrum, then we're able to piece together the pairwise interactions. It takes an enormous amount of time, and I think it's fair to say in our NMR facility, at the moment, we don't really believe that it's reliably automated. A lot of groups are publishing that they've got reliable ways to automate the picking of this. In this case, this was about 7,000 different peaks that are in there, and they're manually assigned. And this is why the process takes several months to work out. But in the end, what you have is an awful lot of information that tells you what the fold of that protein is. The residual dipolar couplings we alluded to before, what we do is we measure the splitting. This is in black is the splitting that we would get for an NH pair in the absence of any adulteration of the medium. So that's just where the D value, this dipolar coupling value, is 0. And the separation there is just J. If we take a collimating medium that allows us to partially orientate the sample so that it has a slight excess population in aligned with the magnetic field, what we'll see is we introduce a very small amount of dipolar coupling, and it's a very manageable amount when we get to this level. So the dipolar coupling here is, if we take, this is J plus D, and in black, it's just the J value. So we subtract the two, and you've got the value of the residual dipolar coupling. That looks really a bit lame unless you see it in practice. So when we take, just as an illustration here, we've got a small 3-helix bundle. If you look at the orientation of these NH bonds here, they align roughly coaxial with the helix axis. So what we would expect is a whole stretch of these residual dipolar coupling values for the NHs that are of similar magnitude because they're similarly aligned with the helix. And actually, when you put the helix boundaries on there, you can see RDCs are really useful for giving us structural information about how different elements of secondary structure are aligned in a molecule. We can do that in challenging systems. So this is a homotetramer. So each subunit is 355 residues. And what we've done in this case, we've got a core domain of the 4 units in the middle, and coming out from there is the first 93 residues. We've done on each of the 4 subunits, we've plotted out 20 candidate structures, and it shows us the way in which this intrinsically disordered tail is held out into solution in this particular case. We get the simulated and the back calculated, sorry, the experimental and the simulated values for the dipolar couplings only agree when we have a single turn of alpha helix at this position in one of the known recognition elements in the N-terminal domain, the intrinsically disordered domain. And we also have 22% polyproline helix here, which helps rigidify the chain and it holds it out from the assembly. This is the sort of information that we can extract. I'm not going to go very much into this other than to say chemical shifts contain some information also on the secondary structural elements. We can identify the presence of secondary structural elements quite easily from the measured values of chemical shifts. The other one that's quite important is used in the same way that fret labels are used in biophysics. We can put a reactive label, a nitroxide spin radical, paramagnetic radical, that we label into a protein at a specific point, and all signals within the influence of that spin, that radical, that is within about 30 angstroms, will be perturbed. Their relaxation will be perturbed by the presence of this powerful paramagnetic spin. If we then add a scorbate, we can reduce this nitroxide into a diamagnetic hydroxyl, and that acts as our control. So in other words, we measure the effects on the spectrum before and after the addition of a scorbate, and that gives us a lot of information in terms of threat labels as what spins are within proximity of this paramagnetic label. So it's called paramagnetic relaxation enhancement. You might be quite familiar with this. It's the way in which we generate structures of proteins. We also use modeling algorithms like this in EM and crystallography. Well, maybe not so much in crystallography, actually. It's the traditional way of doing things. We have a whole set of restraints on the system that we have to optimize at the same time. And we add restraints from the experimental data. Just to recap, it's the NOEs, the residual dipolar coupling, the chemical shifts, what information it's giving us from there. We can use some J coupling constants, and we can identify hydrogen bonds that we can put in as restraints, and paramagnetic relaxation enhancement distances as well. So we put all of that into the system, and what we would expect to see at the end is a structure that has no violations at all from the experimentally measured restraints. And there are very few ways in which you could possibly fold a protein that would give you no violations with the experimental data. It's worth saying, just pointing out, that when you look at you're downloading a crystal structure from a database, you get a single structure. Okay. If you download an NMR structure, what you'll see is an ensemble, an overlay of multiple conformations. So the reason for that is the way in which we determine the solution structure here. What we're trying to do is optimize a coordinate to get the minimum energy. And hopefully, as you do your calculations, you give the system enough energy to get over all of these energy barriers, slowly withdrawing the energy from the system in a simulation, okay, computer simulation. And the whole process is referred to as simulated annealing. And eventually what you would hope to see is that you get into the global minimum. Obviously, in practice, you are simultaneously optimizing coordinates from thousands of different variables that are within the molecule. Okay, so how can you be sure that you have got to the global minimum as opposed to one of these local minima? And the way you do that is you repeat the simulations many times. Anything from 20 to 100 times is typical. And if they all converge to the same point and they are consistent with the experimental restraints, then your confidence that this is the right solution structure goes up enormously. So that's why when you download a structure from an NMR structure from a database, you'll get a multiple overlay, and you can see what is the extent of convergence in the different parts of the molecule from these calculations. AlphaFold has been massive for us in giving us candidate structures, and other than saying that we have the ability to look at the unfolded portions as well as the folded parts. So you may be able to see in there, there's a pink domain, which is the last structure that I did by NMR, and it has a good correspondence, correlates well with what AlphaFold picked out later anyway. But we also have the ability to look at preferred conformations and isolated helices in remoter parts of the molecule. But AlphaFold has been a massive benefit for NMR. If we look at a typical proton NMR spectrum for a protein, you see that it is dominated by the effects of functional group chemistry. So we get clusters of these spins in particular areas. So the NH protons in the backbone of a protein are over here, methyl groups are over here, and the aliphatics from other backbone protons and H alphas are in the middle. Okay. We very rarely work from the one-dimensional spectrum. We work from this one, and it requires us to put in M15. So we label up the protein with the M15 stable isotope, and it gives us the ability to produce these so-called HSQC. It's referred to as a heteronuclear single quantum correlation experiment. and we get a separate spot for each of the NH protons in the molecule. Okay, so it gives us a reporter, a structural reporter, and then we can probe the protein with different binding partners. So in this case, we have now added, we're titrating in a binding partner. We see some of the spins are completely unaffected. They're nowhere near a binding interface, but other spins migrate as we add in more and more ligand. And we can plot those changes, the extent of those, onto the surface of the protein to give us the location of a binding surface. Can put that graphically as to where in the sequence this binding occurs. Obviously, it's not always in a contiguous patch because the protein folds round and other parts of the chain may come into proximity of this binding site. This is all very quantitative. We can then plug in the values of these and we get a standard binding isotherm of the chemical shift perturbation versus ligand concentration. And we can work out with quite high accuracy values of the affinity constants. Here's something where we can look at multiple liganding events at the same time. It's a bit difficult to see. There's coloring on these schemes that is showing what perturbation that we see at each stage. And basically, it's a blood clotting protein in which we've got 3 different ligands that are binding. And we can look at the influence, the structural influence that it has on the protein as this gets perturbed as we add in each of the 3 different ligands. So we can do complex and decipher and delineate complex multi-liganding events. This comes from something that Stefan Freund did several years ago. We can dock molecules together. So this is an unlabeled peptide, and we've got a labeled protein domain, and we had an argument with the referee about whether this was feasible to get this to recognize a binding surface. We've taken a filtered NOE spectrum at this point. What we do on this axis, we reject any signal that is a proton coupled directly to C13. So we only allow through the filter those that are coupled to C12. On this axis, we only accept proton signals if they're coupled directly to C13. In other words, what we're seeing for these cross peaks here is only the signals that originate from the unlabeled peptide and go onto the labeled protein survive the filter. So these are interface, NOEs across an interface only, and all of those within the protein or within the peptide are filtered out. And in this case, we were able to assign 35 different intermolecular NOEs, and we put it into a flexible rigid body docking algorithm called Haddock from the University of Utrecht. And it gives us high accuracy data docked structure for the complex. Higher molecular weight. I'll try to speed this up now. Sorry about this. Slightly overrunning. What is the molecular weight limit for NMR? And it's really hard to say. It depends what information you want. If you want a high resolution structure, the molecular weight limit is around about 40 kilodaltons-ish. But you can still see the influence of adding binding partners, even in quite high molecular weight complexes. We add a potential binding partner, and we can show we are getting perturbation of the system. Getting assignments of each of those signals is quite difficult in this case, but it still provides us with some information. So it's difficult to say in absolute terms what a molecular weight limit is for the sort of information you can get from NMR. We don't need to be fixated on the protein itself. We can also look at, in this case, a very simple one, substrate turnover. This is a DNA translocase from Jan Lerver's group, and we're looking at the rate at which this enzyme turns over ATP, and we can get a lot of information. You possibly can put in inhibitors or whatever to study how this is affected by various processes. But looking at the small molecule is also something that we're receptive to. We can also look at small molecules inside whole cells. This is an NMR spectrum of an intact erythrocyte, human red blood cell. And we see the evidence of broad signals there from macromolecules. There's a broad hump underneath these signals as well. Could it be really so simple that if we delayed the start of acquisition of data so that we didn't acquire any data in the first few milliseconds, we would lose virtually all of the signal from the macromolecules, and we would lose almost nothing from the small molecules. Well, it is that simple. And if we do that so-called relaxation filtered experiment, we can filter out the effects of all of these macromolecules, and we can get high resolution spectra for the small molecule components of the cytosol. And if we look in detail, there are some groups that are looking into this kind of stuff. It's referred to as metabolomics, and you can simultaneously from the one spectrum be monitoring the changes and perturbation of hundreds of different biomarkers within a cell that you can identify. NMR is able to get the molecular weight, get an estimate of molecular weight. It's not a mass spectrometer, it doesn't do that kind of accuracy, but certainly we can tell you whether a molecule is rotating as a monomer, dimer, trimer, or whatever. So we have some information on the effective molecular weight of how these things are tumbling in solution. Built-in QC, built-in quality control check, is a very important part of what we do. This is a spectrum for an unfolded protein. So this is up at the NH region, this is the methyl region. We get no dispersion of methyl signals at all in an unfolded protein. All of those methyl groups are sitting out in solution. They're all in exactly the same environment. As we fold the protein up, those methyl groups experience different environments and they spread out into different frequencies. And so we instantly can tell somebody, is my protein folded or not? Well, within 5 minutes, we can tell somebody, yes, it is or it isn't. And that is not an insignificant amount of information to give to somebody. If you're doing assays and you're struggling, you don't know what condition your protein is in structurally. NMR can give you an idea. There are other techniques, CD or whatever, that could also tell you, but NMR gives you that information very quickly. In the two-dimensional spectrum, we see pretty much the same thing. This is a typical sort of HSQC that we see for a folded protein, for an unfolded protein, and for an aggregated protein. And the feature of the aggregated is that we get big variations in the intensity of these peaks as things clump together. In terms of a quality control check, would we recommend that you go on and continue doing assays when clearly you've got aggregated protein in there in the first place? We would recommend you spend time working out your protein solution conditions before wasting time on further experimentation. Jane will come into this. I really will wrap up now. This is just saying we can encode the amount of flexibility in the backbone of a chain, protein chain, into the intensity of peaks in these HSQCs. So here we plot the peak intensity as a function of residue number, and you can see the first few residues are unstructured in this particular case. This is a very simple example on which we can get dynamic information out. Here's a real case example. It is looking at a 3-domain protein of clinical significance. It is isolated from cancer patients. In this particular case, we can see from the relaxation plot, one domain, which is the red one here, is rotating at a different rate to the other two, which resonate together or rotate together. So there's intrinsic flexibility in that link, and mutations in that region are connected to clinical manifestations. And in this particular case also, we've got a yeast and a human, and you can see there is one element there. It's beta 6, the beta 6 strand. You can see goes to completely different mobility. And it's much more mobile in the yeast homologue. And if you see this beta 6 strand, what it's telling us is that whole strand comes out into solution and it isn't sitting stably inside the fold of that protein in the yeast homologue. So that's the sort of information. It's really difficult to get that with that kind of site-specific detail from any other technique that we know of. We can get, yeah, it's quantitative. We can get biophysical constants and pKa's of individual side chains site-specifically. And yeah, NMR screening, well, it can be done. We can identify from libraries of compounds, we can identify compounds that bind to the protein surface. And don't know what that's doing there. We've had that. That's, yeah, not something. This is a series of experiments that we can use to distinguish. In this case, we've got two small molecules, one which binds to the protein and one which doesn't, and a series of experiments that will discriminate between the behavior of the two in high throughput or moderate to high throughput. So that's the end. It's just a summary slide to say we get a protein quality control check when we do all of our studies, so we know if the protein's gone off. We can do structure and dynamics and molecular interactions. You're looking at protein-protein. Also, we do some nucleic acid work and some small molecule work as well. And we can get out biophysical rate constants.